As we learn more about the DNA in human cells, how it controls the growth and development of the cell. Mm, In the laboratory, they seem to programmed to divide only a certain number of times before they die. Now, this differs with the types of the cell. In some cells, like nerve cells, only divide 7 to 9 times in their total life. Others, like skin cells, will divide many more times. And finally, the cells stop renewing themselves and they die. And in the cells of the human body itself, in the cells of every organ, almost every type of tissues in the body, the same thing will happen eventually. Okay, you know that all of a person's genetic information is contained on very long pieces of DNA called chromosomes. 46 of them are in the human cells, that 23 pairs of these chromosomes are varying and sizes. Now, if you look at this rough drawing of one chromosome is about to divide into two. You see that it looks like, well, actually it's much more complex than this, but it remains as a couple of strings linked together, two called pieces of DNA. And if you stretch them out, you will find they contain certain genes, certain sequence of DNA, to determine how the cells of the body will develop. Researchers look very carefully at the DNA in chromosomes, though. They were made as we all were to find that only a fraction of it, maybe 20 to 30%, convert into meaningful genetic information. It's incredible, at least also to me, but if you took away all the DNA that codes for genes, you still have maybe 70% of the DNA left over. That's the so-called junk DNA, though the word junk is used some tranquility. It was some of the purpose. Anyway, if we examine the ends of the coil of DNA, we find a sequence of DNA at each end of every human chromosome, called a telomere. Now, a telomere is a high repetitious and genetically meaningless sequence of DNA, what we are calling junk DNA. But it doesn't have any important purpose. It's sort of like the plastic tip on each end of a shoelace. It may not help to tie your shoe, but the rest of the shoelace shoestring from unraveling to weak and useless thread. Well, the telomeres at the end of chromosomes seem to do about the same thing, protect the genes, genetically function parts of the chromosome from being damaged. Every time the chromosome divides, every time one cell divides into two pieces of the ends of chromosomes, the telomeres get broken off. So after each division, the telomeres get shorter. One of the things that may happen after a while is that pieces of the genes themselves get broken off the chromosomes. So the chromosome is now losing important genetic information and is no longer functional. But as long as the telomeres are at certain lengths, the activities have happening. So it seems that by looking at the length of the telomeres on a specific chromosome, we can actually predict pretty much how long certain cells can successfully go on dividing. Whereas some cells just seem to keep on dividing regardless, which may not be always a good thing if it gets out of control. Chemically, we learn how there's a chemical in them, and an enzyme called telomerase. As bits of the telomere break off with the end of chromosome, this chemical, this telomerase, can reveal it, can help reassemble the protective DNA, the telomere, that the chromosome is lost. Someday we may be able to take any cell and keep it alive, functioning and reproducing itself essentially forever. In the future, we may have virtually immortal nerve cells and immortal skin cells or whatever because of this chemical telomerase. can keep the telomeres on the ends of chromosomes from getting any shorter.